Good afternoon, everyone. I'm Dirk van HYFT. I'm the CTO of IPA, immunoprecise antibodies. And we are in the development of both diagnostic and therapeutic antibodies. And today, well, it's our great times now, because you really have the fusion of AI and biotech. And so one of the big questions is it really an AI tech push, or is it the other way around, that we can apply AI on biological models? And so I'm really glad that I also have studied medicine and I did a specialization of psychiatry, so that I really also could understand what is the complexity of biology. Because if we want to model biological processes, it's huge, it's multidimensional, and we need to take into account quite a lot of parameters. And so what is really necessary, if we really want to build AI models for biology, that we start from the biological background. Because if we look now at the current AI models, then we see some shortcomings. For instance, the all well-known AlphaFold prediction tools, they are based on homology. So multi-sequence alignment. So first, that means if you want to predict a certain structure, you need to start from a sequence. Then you do a massive lookup in similar sequences. So that's really looking up at homology patterns. And then you can start doing the prediction. So that means it's not going from a zero without any knowledge in advance. No, you need to add this knowledge before you start with your structural prediction. On the other hand, also you see that if you want to, for instance, make antibodies, that you need to integrate quite a lot of data and information really from different disciplines. And so I will give an example during the talk about a serotonin transporter. So that is something that is a transporter in the brain that also can help to elevate depressive symptoms. And if you want to understand that, then you need to look at, okay, where in the brain is this transporter? How is it reacting with certain medicines and so on? So again, it's quite complex. And what you see is that at this moment, all this data reside in different silos. Also, on the other hand, well, we know it's quite a lot about tokens and embeddings, but you see that all these models are very static. So they are trained for a certain purpose, and then, yeah, you can use it for your inference step, but in a certain way, it's very difficult to add new knowledge. And so that's why we have built a very, what we call bio-native approach to address these problems. And so we have found a kind of indexing system based on HYFT patterns. And with this indexing system, we can combine sequential information, structural information, and functional information. So in one big framework, we can integrate all these different modalities. And that helps us also to understand and even integrate information from different large language models. Because on this HYFT patterns, this index, we can integrate information from different large language models that incorporate, for instance, sequential information, but also structural information. And with contrastive learning, we can see, okay, but this and this models are adding extra knowledge to our model, or this is just redundant. And so we can see that our HYFT patterns as a kind of indexing system and stacking up different large language models and help to align information of sequences. So let's look a little bit more to the HYFT patterns itself. Well, at some point, I did a discovery, and I really found that indeed in proteins, so the long sequences of amino acids, there are also called a certain type of words. So really semantic groups, semantic patterns that really make sense in the biology. And so with this 660 million of these patterns, you can index whatever that exists in the biosphere. It goes from plants, it goes from viruses, it comes from yeast. So whatever exists can be indexed with these patterns. And based on that, we have built a kind of, call it RAG system. So that means you want to look, for instance, to a certain sequence, you do this indexing and then parsing to all these HYFT patterns, and then you ask specific questions or do retrieve specific elements out of databases. So it's really, again, a combination of sequential information, structural information, and functional information in a multimodal way. And if we compare it with current methods, like I said already, so for instance, the well-known AlphaFold for the structural prediction, it starts with this multiple sequence alignment. And that's a very compute-intensive process. On the other hand, if we also look at the functional information that can be extracted even from different large language models, then you see that these large language models are also trained for very specific functions. So they are not integrating, let's say, a holistic way of looking at biology. And so we solved that problem. So one of the challenges is indeed making sure if we start building AI models that they are bio-native, so that we really start from the biology, from the complexity of the biology, so that we build bio-native models that understand the biology. And also what is really important to understand, it's really a bottom-up approach. So we started with indexing whatever exists in the biosphere, and from that data on, so the bottom-up approach, we have built our model, and not the top-down approach where you have predefined models. And that means that it's just not one single tool, but it's a platform that integrates all these different modalities and helps us also to understand the context of proteins in certain disease states or whatever. So the fact that we started to build from the biology itself helps us to do some inference, and also to explain that we have a cross-model retrieval system. So we have indexed every sequence in the biosphere. So let's say around 500 million of these sequences. We have structural information. We have information that we extracted also from text. And in that framework, you can start doing reasoning and extracting specific elements that are necessary for the antibody development. An extra challenge that you have if you want to build AI models for biological discovery is to make sure that they are scalable. Again, I referred already to that first multi-sequence alignment step. That's a very intensive process. It takes time to get all your results. And with our approach, with our HYFT-based approach, you can really speed up that process. And that really makes sense. Just to give you one example, for instance, we have built different pipelines in the antibody development. And for instance, one pipeline is looking for immunogenicity and to screen if certain patterns in the antibody are also in the human proteome. And for instance, doing that lookup and to see if there is a similarity between the antibody and the human proteome, that's just a matter of seconds. And otherwise, if you have to do that lookup, it will take really quite a long time. And so you cannot screen it in a massive way. So by using these HYFT patterns, we can speed up the process and do massive analysis on huge data sets. And another important challenge with AI models is that you need to be sure that you can generalize information. And the fact that we have combined from sequence to structure to function in one framework, we can start to understand new relationships. And that helps us also to reveal new elements. And also, I will give you an example of a demo where we started looking at a sequence that is of very special interest for a psychiatrist, namely a serotonin transporter. So that's again a certain protein in the brain, and medication, antidepressants, are working in combination with these transporters. They are blocking this transporter. And in that way, that serotonin still stays at the intersynaptic area. And by blocking this transport, the serotonin cannot go in the neuron. And that helps us to elevate depressive symptoms. So first of all, because I was interested in that sequence, I used our search engine. And here you really see a live demo I've recorded, but I think it's important to see how fast we do this lookup in 440 million sequences. And everyone who is aware, if you want to do this search in a classical BLAST manner, with that application, that will take too much time. And again, it's not integrated with all the databases. And maybe you have seen it at the beginning. We do this search not only at the amino acid level, but also at the DNA and the RNA level in one single, again, framework. And we aggregate all the data and referring to all the different species where this transporter resides. So I was already very intrigued by doing this kind of lookup and such a massive scale, because I could also see that, for instance, this serotonin transporter was also at certain bacteria. And that was really curious for me to see that point, because I always had the idea that this serotonin transporters are in the brain, but the fact that I could see it now in a certain bacterium, helped me to say, okay, here I need to look more in detail. And so, I have to go back to the, so here I did this first lookup. You can see that we also can select the different databases and look for a sequence alignment. And here, and I will pause it, that is that we not only have the description of each sequence, but also what we call the top concepts. So we have parsed all the descriptions in meaningful concept, and that means also that we can do a semantic analysis of the returned results and connect that with the information that is in the literature. So the next step, once we have found our protein, we also want to look to the 3D structure of that protein. And here we have integrated this with an agentic AI where we can ask specific questions. And for instance, one of the questions that we ask here in this transport is, okay, assign the specific amino acid that is responsible for the stabilization of this molecule, and that also fits with this specific drug. And you see by asking questions, color first that specific amino acid, and then also the drug, as have a better understanding of this molecule. It's not only a matter of selecting amino acids, but I can also ask to calculate certain physicochemical properties. And for instance, one of the properties for an antibody is the hydrophobicity. And here I can ask the question, okay, calculate this this physicochemical property, and then immediately the calculations went on. So once we have now looked from sequence to structure, now we want also to look to the functions of these proteins. And here we can activate what we have stored in our knowledge database. So we have indexed the whole PubMed, so all the PubMed abstracts, but also full articles. And we parse this in concept relation concept pattern. So meaningful patterns, not just words, but meaningful patterns. And this concept relation concept patterns is also referred to predicates. And that's the way that we as human beings are reasoning. And I just want to show you how easily we can extract these reasoning patterns. And so you see that, for instance, the serotonin transporter is in combination with different other concepts, how it is influenced, where is it located, and so on. So it's really a way to extract in a massive way this knowledge and store that in our knowledge base. And with this, we have an interface that we can start asking questions and make this very relevant. So after looking at the CRC patterns, we can also look at what are all the other concepts that are in the neighborhood of my serotonin transporter. And that helps also to understand the broader context of what is described about this specific molecule. And for instance, you could see that certain adverse events are mentioned or specific locations. And again, the fact that we not just take words, but meaningful concepts give us a really in-depth understanding of the biology of this transporter. And of course, yeah, with all these agentic technologies, you can also ask, okay, I want to use specific large language models or reasoning models to further analyze this data. So in a nutshell, I started from a sequence. I did this search in a massive way, so more than 440 million sequences in seconds. It was integrating DNA, RNA, and amino acids at that different level, even spread over different databases. Then I could see, okay, these are the descriptions, but also the meaningful word groups, these concepts. This gives me an anchor point to functional information that resides in textual data. And I also could start analyzing the structure and asking questions to that structure and say, select me this of this amino acid, or make some calculations on certain physicochemical properties. So yeah, we are excited to be here, of course. First of all, as we all know, yeah, NVIDIA has the huge power, the computational power that we need to make all these tokens and to have the integrations of our stack of the different large language models associated with our HYFT patterns. So that's of course nice, but you also, it's not just the hardware, you need to have it in the cloud. And then that's why we are also very proud that yeah, we're working together with Vultr. And again, it are not just machines, but there are real people that also help us to yeah, accomplish our goals. So together with Vultr now, we start to bridge the biology and the AI through computational methods. So thank you very much for your attention.